Welcome back to the deep dive. We are your guides through the often complex world of research.
And our mission is pretty simple.
We take a stack of dense, technical sources and we distill them down.
Right, into the nuggets of knowledge you need to be, thoroughly and quickly informed.
Today, we are wading deep into a topic that has, I think it's fair to say, moved far beyond just being a health food trend. We're talking about kefir.
We are. And for this deep dive, we're not just looking at it as a trendy fermented drink.
Yeah.
We're viewing it as an incredibly complex, living biological system. It's something scientists are really intensely investigating for its molecular potential in long-term health management.
That's right. Our mission today is laser-focused. We've stacked up systematic reviews and meta-analyses.
The highest level of evidence out there.
Exactly. And we're using them to uncover the specific isolated components within kefir. We're talking about its molecular toolbox.
We're identifying these powerful bioactive compounds.
And we're mapping out exactly what the robust science says they can do.
Particularly when it comes to managing these silent threats. Things like inflammation, metabolic syndrome, and high blood pressure.
All major long-duration health concerns.
And to really set the stage for why understanding these molecules is so critical, we have to talk about the primary enemy they seem to be designed to fight.
Which is?
Chronic inflammation. It is the silent driver behind so many of the chronic diseases we face today.
Okay, so the key marker we need to track, the smoke alarm in your body, is something called C-reactive protein, or CRP.
That's the one.
Let's unpack this a bit. So CRP is more than just a vague sign that you're sick right now. You're saying it actually predicts future problems.
It does.
What is it exactly? And what is its role in long-term cardiovascular damage?
Okay, so CRP is a key protein produced mainly by the liver.
Mainly the liver.
But also, and this is really significant, by adipocytes.
Your fat cells.
Exactly, your fat cells. And this happens in response to stress signals in the body. We know from a lot of data that an elevated level of high-sensitivity CRP is a serious independent predictor of future cardiovascular events.
So it's not just a correlation. It's a measurable risk factor.
It is. It actively predicts future cardiovascular events. But the interesting part is how this inflammatory mediator actually messes with the system that controls our blood pressure.
Okay, how does it do that?
Well, this is where it gets really interesting in the context of long-term health and managing blood pressure. As an inflammatory mediator, CRP doesn't just sit there. It actively interferes with the body's very sophisticated blood pressure regulation systems, primarily by stimulating the renin-angiotensin-aldosterone cascade.
The renin-angiotensin cascade. That's the system we often hear about with hypertension, right? Fluid retention, sodium.
Correct. And by stimulating that cascade, CRP enhances what are known as pro-atherogenic effects.
Okay, break that down for us. Pro-atherogenic.
It means it actively promotes the hardening of the arteries, or atherosclerosis. More specifically, it causes these structural and functional alterations in the vascular wall.
So in the walls of our blood vessels.
Yes. It leads to vascular thickening and stiffness. In short, it's a crucial biochemical link between that low-grade chronic inflammation and the slow evolution of hypertension over years or even decades.
So if kefir can reliably intervene in this chronic inflammatory process,
then we are talking about a powerful and really accessible tool for preventing long-term cardiovascular damage before it ever becomes a crisis.
that really grounds our deep dive. We're looking for molecules that can interrupt that cascading path to chronic illness.
Precisely.
So let's start at the source. Let's decode the kefir grain itself. This is a traditional fermented milk, and it's created using a starter culture that comes from these complex grains.
The kefir grain is, it's truly a biological marvel.
It's not just a powder you add.
No, not at all. It's not just a culture. It's a complex, self-sustaining, symbiotic ecosystem. It's often referred to as a SCFA.
A SCOBY, like with kombucha. Symbiotic culture of bacteria and yeast.
That's it. It hosts this incredibly diverse range of microorganisms, all living together in a very strong, specific relationship that, it maintains the structure of the grain itself.
What exactly makes up this microbial diversity? Is it all just bacteria, or is the complexity spread out more?
No, and that's the fascinating part that makes kefir functionally so different from, say, yogurt.
Okay.
The core includes various species of beneficial bacteria, like the essential Lactobacillus kefiri, and you've got species from the Leuconostoc, Lactococcus, and Acetobacter genera.
So a lot of bacteria.
A lot. But crucially, it also includes a rich community of yeasts and fungi.
Oh,
You
okay.
have lactose fermenting yeasts like Clevermyces marxianus, and then you have non-lactose fermenting yeasts like Saccharomyces unisporus and Saccharomyces cerevisiae.
The baker's yeast.
The very same. This rich abundance of bacteria, yeasts, and fungi all working together in this grain matrix is what sets kefir apart and generates its very specific molecular output.
It sounds like a tremendously rich and cooperative microbial environment.
Yeah.
And it's this intense fermentation that generates the molecular toolbox we're interested in.
That's right.
When we isolate the molecules that are responsible for these long-term health benefits, what are the four key heavy hitters?
There are four key components that are consistently identified in the literature. First, you have kefiran.
Kefirin.
Kefiran is actually the physical backbone of the grain structure. It's a water-soluble polysaccharide, so it's a sugar structure, and it's categorized as an hexapolysaccharide, or EPS.
Okay, so EPS is number one. What's next on the list?
Second, we have bioactive peptides.
Peptides, right.
These are small fragments of milk proteins, mostly caseins, that are released and crucially activated during the intense fermentation process. The microbes are essentially breaking down these large protein structures into smaller, more powerful pieces.
Okay, so we have EPS and peptides. What's the third?
Third, you have organic acids, especially lactic acid.
And that's what gives it that sour taste.
It is. It gives kefir its characteristic sour taste and also its preservation qualities. It's a major byproduct of the fermentation.
And the fourth component, the one that gives kefir its competitive edge in that little microbial world.
That would be the bacteriocins.
Bacteriocins.
These are proteinaceous toxins that are secreted by the bacteria, and they are specifically designed to inhibit the growth of competing and often pathogenic strains of bacteria.
So they're like microbial assassins.
You can think of them that way, yes. They provide a competitive advantage within that complex grain structure, keeping it clean and stable.
So when we talk about kefir providing long-term health benefits, we are really talking about the delivery system for this potent toolbox of molecules.
Exactly. Kefiran, peptides, organic acids, and bacteriocins.
And for you, the listener, to accurately interpret the efficacy of the studies we're about to dive into, you have to note a key distinction.
Yes, this is critical. It's how the kefir is produced, because that dramatically impacts the functional potential of that molecular toolbox.
Okay, what are the two main types?
You have two primary forms that are studied. First, artisanal kefir. This is produced using the traditional kefir grains we just talked about.
So it has the maximum microbiological diversity.
And crucially, the resulting molecular output. Then you have industrial kefir.
The kind you see most often in the supermarket.
Right. And that's produced using previously selected controlled starter cultures. As we'll see later, that difference in microbial diversity, it matters enormously, especially for functional components like antioxidants.
Okay, that's a huge point to keep in mind. Let's shift gears now from the molecules to the highest level of proof, the clinical evidence. We're now looking at systematic reviews of randomized controlled trials, or RCTs, which assess these effects in humans.
Right. And when you start reviewing these human trials, you immediately run into a significant challenge.
Which is?
Heterogeneity.
Meaning everything is different.
Everything. Major differences in dosage, the duration of the intervention, the type of control groups used. I mean, some used a placebo, some used unfermented milk, others used different dairy products, and of course the patient's existing health status was all over the map.
That makes it really hard to compare apples to apples.
Very. Despite this, the most rigorous meta-analysis we looked at managed to pull together 7 relevant RCTs for a focused look at blood pressure effects.
So what did those studies look like? Who were the participants? And how long did the interventions last? This is really crucial for our focus on the benefits of long-term consumption.
The participant profiles were quite diverse, which actually reflects kefir's potential applications across multiple chronic disease states.
Like who was in them?
They included healthy adults, individuals with metabolic syndrome, people with inflammatory conditions like ulcerative colitis and Crohn's disease, non-alcoholic fatty liver disease, or NAFLD, and even some COVID-19 patients.
Wow, so a very broad range. What about the dosage and duration?
The dosages ranged significantly, from a minimum of 90 millimollo up to 500 millimoles per day.
So from less than half a cup to over 2 cups.
Right. And critically, the duration of the intervention, or our long duration factor, only varied between 2 and 12 weeks.
12 weeks. That's really the upper limit we're seeing in this high-quality human analysis.
It is.
That means the observed benefits, if there are any, are achieved in relatively short intervention periods. But it also means we still lack solid, high-level data on what happens after, say, 6 months or a year of consistent use.
Absolutely. That lack of true long-duration data is a limitation we absolutely have to flag for everyone.
No doubt. That said, let's look at the hard data for blood pressure from these trials.
Okay, so the analysis assessed the impact on both systolic blood pressure, SBP, that's the pressure when the heart beats, and diastolic blood pressure, DBP, the pressure when the heart is at rest.
And for SBT, the top number.
For SBP, the overall effect, measured as the weighted mean difference, was a non-significant reduction of net of 1.76 ml/Hg.
Okay, so on average, a small drop in the SBP number,
Yeah.
but not statistically reliable across all those diverse studies.
Correct. It didn't reach statistical significance. But here's the key DBP takeaway.
A bottom number.
Yes. Kefir consumption was associated with a statistically significant overall reduction in DBP. The weighted mean difference, the WMD, was -0.47 ml/Hg, and the p-value was 0.04.
Okay, let's pause on that. A WMD of -0.47 mmHg.
Yes.
To a lot of people, that sounds, well, negligible. It sounds like a tiny reduction. Why is that statistically reliable finding still compelling for long-term health?
That's an essential question for interpreting meta-analyses like this. The weighted mean difference is the result of averaging the impact across all these different studies, and it compensates for their varying sample sizes and rigor.
Okay.
And while 0.47 mmHg sounds numerically tiny, the fact that this subtle reduction is statistically reliable across 7 diverse, independently conducted trials, that's the real news.
Meaning it's very unlikely to be due to random chance.
Exactly. For a simple dietary change, achieving a reliable, persistent reduction in DBP, the resting pressure in your arteries, is actually very compelling for managing chronic cardiovascular risk over an entire lifespan.
That reframes the data completely. It's the reliability that matters most for long-term prevention, not just the size of the number in a short study.
Precisely.
What also stands out is the subgroup analysis you mentioned, the one on age.
Yes. This is where we see how demographics can influence results, and it might even link the outcome back to that chronic inflammation we talked about at the start.
What'd they find?
When the researchers looked closer, the DBP reduction was notably stronger in the age group greater than 45 years old.
Compared to the younger group.
Right. The p-value for the over 45 group was 0.31 compared to the younger group 45 or less, where it was 0.81.
So it suggests that the beneficial effect on diastolic blood pressure may be more pronounced or maybe just more relevant in older populations.
It does. Perhaps in those who are already experiencing some degree of that age-related vascular stiffening or the inflammatory interference caused by elevated CRP we discussed.
So it hints that as inflammation becomes a more chronic factor, as we age, the anti-inflammatory mechanisms of kefir might kick in harder or just have a greater impact on those already compromised systems.
That's a very plausible hypothesis.
Now let's talk about the quality of these human trials. Because a reduction is only as reliable as the study that produces it.
And we must apply extreme caution here. While the overall quality score of the RCTs was rated as moderate, the analysis, using the Cochrane Collaborations tool, revealed that many trials had a high risk of bias.
In what areas specifically?
Specifically, major issues were identified regarding the lack of blinding of participants and, critically, the blinding of the outcome assessors.
Okay, why is the blinding of the assessor such a critical gap, particularly in a functional food study like this?
Well, if the researchers who are measuring the blood pressure, for instance, know whether the participant is consuming the active kefir or a placebo,
Their expectations could influence the result.
even subconsciously, yes, it can potentially influence how measurements are taken or interpreted. This introduces what's called observer expectation bias, and it makes the final numerical result less trustworthy.
And you combine that with the huge variation in control groups.
Exactly. Some used a placebo, some used unfermented milk, others used different dairy products. It makes it incredibly difficult to isolate the true specific effect of the bioactive compounds versus, say, just drinking a non-kefir fermented product or just consuming more dairy.
So the bottom line is that these limitations emphasize that while the initial statistically significant findings on DDP are promising for a chronic condition like hypertension,
We urgently need more long-term, well-designed and rigorously blinded RCTs to really confirm kefir's role as a reliable therapeutic strategy over many months or years.
Okay, so moving from the human trials, which tell us what happens in short time frames, let's transition now to the preclinical research.
The in vitro and animal models.
Right, the studies that truly explore how kefir works detailing those specific compounds and their mechanisms. This is really the heart of the molecular deep dive.
It is.
Let's start with its function as a kind of molecular shield. Its antimicrobial activity.
This power is attributed primarily to three components in that toolbox we mentioned.
Kefirin, organic acids, and peptides.
That's right. The microorganisms in the grain produce these compounds mainly to maintain their own ecosystem and inhibit competitors, and we, the consumers, get to reap the benefits.
What's particularly fascinating here, and a crucial nugget of knowledge for any kind of future pharmaceutical application, is the comparison of potency.
Yes. They aren't all equally strong, even when they're targeting the same pathogens.
How different are we talking?
When you compare interventions against similar target pathogens like E. coli or S. aureus, the research reveals a significant difference in efficacy. The concentrations and intervention times required for the organic acids, like lactic acid, were dramatically higher.
Dramatically.
The concentrations needed were up to 40 times higher.
40 times.
Yes, and the intervention times were twice as long compared to interventions using isolated kefiran or those bioactive peptides for similar antimicrobial results.
That magnitude of difference is huge. It suggests that the organic acids act as a sort of broad ambient inhibitor, just lowering the pH.
While the peptides in kefiran are the high-potency, targeted precision weapons.
So how do they work at the molecular level? What are they actually doing to the pathogen?
The mechanisms are diverse and complex. The bioactive peptides, for example, primarily work by damaging the outer and inner cell membrane of the pathogen.
They punch holes in it.
In a way, yes. This causes a leakage of crucial intracellular components, like potassium ions, which destabilizes the pathogen and kills it.
That's not all they do.
No. Furthermore, these peptides have been shown to bind directly to the pathogen's DNA, which effectively disrupts its ability to replicate and sustain itself.
And the organic acids.
They perform that role of broad-spectrum inhibition. They reduce the overall biological activity of pathogens, and, importantly, they inhibit the germination of spores, such as those from B. cereus. And this effect is entirely dependent on the dose used.
So the meta-analysis of all these preclinical antimicrobial studies, it corroborates that these mechanisms actually translate to a measurable effect.
It does. A statistically significant overall antimicrobial effect was confirmed across the studies. It was specifically strong against fungi and Pseudomonas B.
But less so against others.
Well, while they showed a generalized tendency to inhibit other bacteria like E. coli and Salmonella, the highly significant, most reliable effects were against fungi and the opportunistic pathogen Pseudomonas.
That's a specific and powerful set of targets for long-term defense. Now let's transition to the second key function. Antioxidant activity.
Right. If chronic inflammation is the smoke, oxidative stress is often the underlying fire that fuels it over the long run.
And oxidative stress caused by excessive free radicals contributes directly to vascular damage and aging.
Indeed. And the main compounds involved in neutralizing that fire are the exopolysaccharides, that's the EPS, especially kefirin,
Along with the peptides.
and various phenolic compounds. They are crucial for protecting sensitive macromolecules like our proteins and our DNA from damage.
And we have specific data on that level of protection, don't we?
We do. In laboratory settings, EPS proved remarkably effective at protecting protein integrity.
How effective?
Low concentrations of kefirin, ranging from just 0.05% to 0.25%, were able to protect BSA protein, which is a standard model protein, from oxidation induced by a very harsh oxidizing agent.
And the protection level.
This decrease in protein oxidation ranged from an impressive 31% all the way up to 96% protection.
96%, that's incredible.
It is. The EPS also conferred resistance to hydrogen peroxide, which essentially protects the kefir's own sensitive microorganisms from damage. That's a great indicator of its potency.
So how does the EPS molecule achieve this level of protection against free radicals? What's the mechanism?
It works primarily through two defined mechanisms. First is metal chelating activity.
Chelating, so it binds things up.
It binds up metal ions, like iron or copper, that can otherwise catalyze or speed up the formation of damaging free radicals in the body.
And the second mechanism?
Second, it works by directly sequestering or capturing highly reactive hydroxyl and superoxide radicals. It neutralizes them before they can attack surrounding tissues.
That makes perfect sense. It disarms the raw materials for damage, and it neutralizes the active agents of damage.
Precisely.
But you also mentioned a really interesting biological twist related to the source of the milk, something you call the matrix effect.
Yes, the matrix effect. This was a fascinating finding. The research found that the antioxidant activity differed significantly depending on the milk matrix.
For example, sheep's milk kefir versus cow's milk kefir,
Exactly. And this difference is largely believed to be associated with variations in the protein precursors that are present in the original milk.
meaning the building blocks are different.
Right. Cow's milk has a different balance of caseins and whey proteins compared to sheep's milk. And since these proteins are the raw material for those bioactive peptides,
The final array and concentration of antioxidant peptides produced by the microbes will be different.
exactly.
So the source material, literally, the diet of the cow or the sheep, dictates the composition of the therapeutic molecules that are created during fermentation.
It does.
This highlights a fascinating challenge for standardization. It means that to create a truly potent and consistent therapeutic food for long-term use, you'd need to standardize not just the microbes in the grain, but the raw input material as well.
It's a huge challenge, and it brings us to one of the most complex and arguably most important roles of Kefir's molecules, particularly for chronic disease management.
Which is?
Immune response modulation. This is not a simple switch-on, switch-off system. It's more like a dynamic concentration toggle.
Okay, a concentration toggle. Let's unpack this duality. How can the same compounds be both anti-inflammatory, which is what you want for long-term health, and potentially immunostimulatory, which you might want during an acute infection?
It depends entirely on two factors. The environmental context, so the presence or absence of an inflammatory insult, and critically, the dosage or concentration of the active molecule.
Okay, let's start with the anti-inflammatory role. Let's assume there is already an inflammatory disease model present, which is common in things like metabolic syndrome or IBD.
Right. In models of acute or chronic inflammatory disease, compounds like EPS, the extracellular vesicles released by the microbes, and lactate all play a powerful anti-inflammatory role.
And their primary mechanism is inhibiting the NF-kappa dollar pathway.
That's the one.
The NF-kappa B pathway. What is that?
You can think of it as the body's central switchboard for initiating inflammation.
Okay.
By inhibiting this pathway, the Kefir compounds essentially turn down the volume on the systemic inflammatory response. But they also work locally. By promoting the integrity of the intestinal barrier, they increase the expression of key occlusion proteins like occludin, ZO1, and claudin-1.
So the occlusion proteins, they're essentially the specialized mortar between the bricks of our intestinal wall, right?
That's a perfect analogy.
If that barrier is tight, you prevent foreign substances from leaking into the bloodstream and triggering that systemic chronic inflammation.
Precisely. A leaky gut barrier is a direct contributor to chronic systemic inflammation. By repairing and strengthening this barrier, Kefir compounds provide a long-term defense against unnecessary immune activation.
And there's a pretty spectacular anecdote on the potency of this effect.
Yes, there is. In rats with cotton-induced abdominal granulomas, which is a validated model of localized chronic inflammation, kefiran reduced the granulomas by 44%.
24%, and that's comparable to some serious medication, isn't it?
It is. This effect was considered comparable to the results achieved using the powerful corticosteroid medication, dexamethasone. This evidence truly reinforces the therapeutic anti-inflammatory potential of these natural compounds, particularly for long-duration conditions where that chronic low-grade inflammation is a persistent factor.
Okay, so that's the anti-inflammatory side. Now, what happens when there is no inflammatory insult and the compound switches to an immunostimulatory role? The role you'd want for fighting an acute infection or maybe for post-vaccination support?
This is where the dosage becomes that critical toggle, and the distinction is dramatic.
How dramatic?
In immunostimulatory assays, so studies intended to boost the immune system, the concentration of EPS required was dramatically higher. For example, immunostimulating assays used EPS concentrations ranging up to 5,000 micrograms per milliliter.
5,000. And the anti-inflammatory assays?
They maxed out at only 100 micrograms per milliliter.
That is a 50-fold difference. A massive dosage increase is required to switch the mechanism from fighting inflammation to actively boosting the immune system.
Exactly. And at these higher non-inflammatory doses, the compounds stimulate key immune cells' macrophages, encouraging their activity, their proliferation, and the robust secretion of key signaling cytokines like TNF-alpha, IL-6, IL-1 beta, and IL-12.
So you're turning the army on.
You are. This level of immune stimulation is useful for a better prognosis of infectious conditions, but it really underscores the precision required. The concentration of EPS is the primary determining factor in the role it will play.
So for long-term chronic disease management, where the goal is often a subtle, persistent anti-inflammatory effect.
Controlling that dose is paramount to avoid inadvertently triggering inflammation.
Right. Moving on to another critical area of long-term health maintenance, let's discuss the anti-cancer effect of these compounds. This also seems highly dependent on dosage and, importantly, specificity.
Yes. The key compound here is again the EPS, particularly kefiran, although the peptides also showed some real promise.
And the mechanism.
It involves compelling damaged cells to undergo programmed cell death, or apoptosis.
How do they do that?
They achieve this by upregulating key pro-apoptotic genes, things like cytoshes, BAD, and BAX, which trigger the cell's internal death signal.
And at the same time?
They simultaneously downregulate a gene called BCL2, which is involved in maintaining mitochondrial stability and preventing cell death. So essentially, they tell the cancer cell to self-destruct through the mitochondrial pathway.
The data showing a reduction in cancer cell viability for breast, colon, and cervical cancer models is obviously very promising. But here we run into a serious safety issue, which links directly back to that dosage control we just discussed.
We must detail this safety margin issue. It's a major concern when thinking about long-term or widespread therapeutic application. While high concentrations kill cancer cells, they can also affect normal tissues.
And we have clear evidence of this.
We do. Developmental toxicity was clearly observed in zebrafish embryos for kefiran concentrations above 100 micrograms per milliliter.
And that toxicity gets rapidly worse as the dose increases.
Yes. At 1,000 micrograms per milliliter, which is still within the concentration range, tested in some of those in vitro cancer assays, kefiran resulted in 80% mortality in the embryos.
Wow.
This implies that while EPS is potent, it can affect the growth of normal, rapidly dividing tissues depending on the concentration used. This means we need extreme caution for any clinical application, especially considering long-term high-dose ingestion.
So we have a potent anti-cancer compound, but one that is also toxic to normal development at higher concentrations. Does the other key bioactive component, the peptides, offer a safer, more specific route for long-term supportive therapy?
It appears so, and this is a massive functional differentiator between the two.
What did the research show?
When we contrast EPS with the bioactive peptides, the peptides show a distinct advantage in specificity. In breast cancer models, these peptides successfully reduce the cancer cell number by up to 88%.
88%.
While crucially showing no anti-proliferative effect on normal human mammary epithelial cells at the exact same dose.
So it killed the cancer cells but left the normal cells alone.
It did. And non-fermented milk, conversely, actually stimulated cancer cell proliferation, which just reinforces the profound specificity that fermentation provides to these peptides.
That is a huge insight for developing functional foods. It makes the peptides a much safer candidate for long-term supportive therapy where toxicity to normal tissue has to be avoided.
Absolutely.
Let's pivot now to the gut microbiota modulation, another pillar of long-term health.
The primary mechanism of action here rests with the exopolysaccharides, EPS, including kefiran. They act as prebiotics.
Fuel for beneficial gut bacteria.
The best kind of fuel. Kefiran proved to be an exceptionally good carbon source, significantly better than common sugars like glucose, for the growth of beneficial strains like Bifidobacterium bifidum.
How much better?
It increased the growth of this specific crucial strain by a factor ranging from 20% all the way up to an astonishing 700%.
700%. That's a dramatic increase. It suggests kefiran is more than just a general fermentable fiber.
It's a targeted fuel source that specifically benefits key health-promoting strains.
What does this modulation do to the overall bacterial landscape in the gut?
At the phylum level, which is a sort of high-level view of the bacterial community, both EPS and the bioactive peptides commonly improve the firmicutes to bacteroidates ratio.
Which is a key metric often associated with optimal metabolic health.
It is. And they also reduced pathogenic genera, such as Klebsiella and Escherichia.
Is there a specific link here to hormonal health, particularly for aging populations?
Yes, and this is a fascinating example of targeted intervention. Bioactive peptides were shown to partially reverse the detrimental gut microbiota effects caused by oophorectomy in mice.
Okay, just to clarify for our listeners, oophorectomy is the standard medical model that's used to simulate the effects of estrogen deficiency.
Exactly. It's similar to what occurs during postmenopausal bone loss and the metabolic shifts that come with it.
So what happened in these estrogen-deficient mice?
The peptides notably reduced potentially pathogenic bacteria like Rhambutia and Streptococcus, which have been directly linked to obesity and metabolic dysfunction.
So in essence, the peptides helped enrich the good bacteria and decrease the potentially harmful pathogens in females who are experiencing these hormone-related shifts.
Precisely.
That is a highly specific application. It demonstrates that these kefir-derived compounds can intervene directly in hormone-related metabolic shifts, which is critical for aging female populations and, importantly, for long-term bone health.
Which brings us to osteoporosis.
Right.
Let's look at that.
This is a remarkable area of study showcasing the true peptide power. Using those same overrectomized mice, the standard model for postmenopausal osteoporosis bioactive peptides significantly inhibited bone loss.
How significantly?
The administration of these peptides increased bone mineral density by 41% and increased the structural support tissue, the trabecular bone volume, by an astounding 264%.
264%. Those numbers are extremely high. That suggests not just prevention of further loss, but a dramatic reversal of existing damage.
It does.
How did that compare to standard treatments, like just taking calcium? Well, the results achieved with the bioactive peptides were similar to those seen in the non-overrectomized control group.
Meaning they essentially reversed the bone loss caused by the estrogen deficiency.
They did, and crucially, the outcome was superior to standard calcium carbonate administration alone. The peptides achieved results that were 0.7 times better than the calcium treatment.
So nearly twice as good.
It again underscores that these bioactive peptides are not just supplementary agents. They are extremely potent therapeutic molecules for conditions that really impact long-term quality of life and mobility.
We've established that kefir contains this molecular toolbox of incredibly potent compounds that operate via diverse, complex mechanisms. So now let's address the elephant in the room that plagues all functional food research.
Variability and standardization.
Exactly. Why is it so difficult to confirm these highly specific long-term effects globally?
The primary challenge lies in the inherent variability of the product itself. Remember our distinction between artisanal and industrial kefir. The quality and, more importantly, the type and concentration of the bioactive compounds vary wildly based on the starter culture that's used.
And this difference has been quantified at a shocking level, particularly regarding the antioxidant benefits, which are so essential for long-term protection against cellular aging.
This is the artisanal advantage. The meta-analysis comparing industrial versus artisanal kefir found that artisanal kefir had a 756% higher chance of having measurable antioxidant effects.
756%.
Yes. That yielded an odds ratio of 8.56.
That is not a marginal difference.
Yeah.
That suggests the industrial product is fundamentally missing something critical. Can we pin down exactly what molecule is responsible for that colossal gap?
It boils down once again to microbial diversity and the resulting molecular complexity.
So the traditional grains.
Artisanal kefir, made with those traditional grains, contains a greater diversity of bioactive antioxidant compounds. You get a full spectrum of phenolic compounds, a richer variety of peptides, and that highly potent EPS, or kefiran.
And the industrial kind.
Industrial kefir, however, often relies primarily on a limited spectrum of phenolic compounds. And while those phenolic compounds in industrial kefir may have some reducing power, they were found to be largely ineffective in terms of true radical scavenging ability.
Unlike the complex profile in the artisanal versions.
Exactly. The mix of phenolics and peptides in the artisanal preparations is far more potent.
So if you're seeking the maximal functional health benefit, especially that long-term protection against oxidative stress and the chronic diseases it causes, the traditional method that preserves microbial diversity is overwhelmingly superior.
Because it produces the full molecular toolkit.
Right. But it's important to note, though, this massive difference was specific to antioxidant efficacy.
Yes. No significant difference was found between the two types regarding their antimicrobial potential. So the simpler functions are retained, but the complex, subtle, long-term benefits seem to be lost in many industrial processes.
Beyond production variability, we also need to critique the preclinical studies that gave us all those fascinating mechanisms in Section 3. Where are the methodological gaps in that foundational research that limit their translational relevance to human long-term health?
The preclinical animal models, while they're crucial for understanding mechanism, they present several inherent limitations.
Why?
We noted a major sex bias. 82% of the studies we reviewed used male rodents.
82%.
Yes. Which limits the applicability of the findings, particularly for complex female health issues like osteoporosis and hormone-related metabolic syndrome, which we just found such exciting data on.
And there's an age bias, too. That limits its relevance to the adult and aging human body, which is precisely the population most concerned with long-term chronic disease.
Exactly. A substantial 79.17% of the mice used were younger than 8 weeks old. If you're studying chronic diseases and long-term health maintenance, the very conditions Kefir is meant to treat, using young, healthy animals limits the direct applicability of the findings to adult or aging models.
Where chronic inflammation and metabolic syndrome are already prevalent.
Right. The response of a young, healthy gut is vastly different from that of an aging, inflamed one.
And we see the same risk of bias issues that plagued the human trials also show up in these animal studies.
We did. We observed a high or unclear risk of bias due to a lack of rigor, specifically regarding the random housing of animals, blinded care during the intervention, and, crucially, blinded outcome assessment.
That failure to blind the assessors again.
It means that observer expectations could potentially influence the reported size of the effects, making the results less robust. These gaps really emphasize that while Kefir is an incredibly promising therapeutic agent, the foundational science still requires greater standardization and methodological rigor to fully confirm its role in long-duration health.
Let's bring this back to a real-world, highly practical context. Let's look at the socioeconomic importance of Kefir, particularly in regions like Latin America, as detailed in the reviews. It's not just a novelty product there, it's an economic necessity for some.
The relevance here is profound. Kefir offers a unique, cost-effective advantage for disease prevention and management, especially in populations where standard medicinal treatments for chronic diseases like hypertension or diabetes are financially prohibitive.
And in Latin America, the prevalence of chronic non-communicable diseases is high.
It is, and pharmaceutical treatments can really strain household incomes.
But Kefir circumvents that financial barrier entirely.
It does. Kefir can be produced by the consumer themselves using grains that are passed down or donated within the community. This significantly reduces the cost of production and dramatically increases accessibility across all socioeconomic strata.
So it serves two markets.
In a way, yes. For those in high-income demographics, it aligns with the trend towards seeking healthier, more nutritious functional foods. But for the broader population, it offers an accessible, sustainable route to proactive, long-term disease prevention that doesn't rely on expensive imported pharmaceuticals.
And the market value certainly reflects this dual health and economic potential.
It does. Even back in 2016, the Kefir market in Latin America was substantial, valued at over $150 million.
Which really underscores its potential for managing these long-term, chronic, non-communicable diseases effectively and affordably.
To summarize this extensive deep dive, we've really moved beyond viewing Kefir as just a simple probiotic drink.
Right.
We're appreciating its complex molecular toolkit, which is dominated by these powerful peptides and kefiran. These bioactive compounds exert three main roles that are crucial for long-term health.
What are they?
First, a potent anti-inflammatory action, but this action is highly dependent on controlling the dosage to avoid that immunostimulatory effect.
Okay, number one.
Second, powerful gut modulation, with compounds like kefiran acting as a highly effective targeted food source for beneficial strains like bifidobacterium.
And third.
And finally, demonstrated positive effects on specific metabolic and structural parameters. This includes a statistically significant, reliable reduction in diastolic blood pressure and that dramatic reversal of bone loss in animal models of osteoporosis that was superior to calcium administration.
The story of Kefir is clearly one of molecular precision, diversity, and duration. We've seen time and again that these small molecular variations translate to massive functional differences.
Peptides are highly potent and specific.
The anti-inflammatory or immunostimulatory effect is controlled by a 50-fold concentration switch.
And the artisanal diversity provides a 756% higher chance of receiving antioxidant benefit than industrial versions.
All these factors point toward Kefir being a powerful therapeutic food, but only if we can control the output.
This reliance on precise concentrations and specific production methods is the future challenge the industry must address. It's how we move kefir from a health supplement into a reliable, long-term therapeutic tool.
Which brings us to our final provocative thought for you to contemplate, linking back to our focus on long-duration health. Given the high potency of these isolated bioactive compounds, the demonstrated functional superiority of artisanal products, and the critical need to control concentration to avoid toxicity or to switch the immune response, what specific ethical and technological standards must the food industry develop to provide a truly standardized, safe, and effective long-term therapeutic food that utilizes the full potential of these complex bioactive molecules?
Is it enough to simply measure live bacteria?
Or must future regulations focus on quantifying the specific potent molecular output, such as the kefiran and bioactive peptide profiles, for both consumer safety and efficacy?